Hello everyone. Today we are going to see about an important topic in microbiology, which is basics in culture media. In microbiology, culture media plays a very big role in the growing of microorganisms. So culture in the sense the growing of microorganisms, and the media, several media is used for the growth of microorganisms. So today we will see about what is media, what are the components, and what are the types of culture media. Yes, culture media, it is often referred to as growth media. So which is in any form of substances like solid, liquid and semi-solid substance, and that mainly aids in the proliferation of bacterial populations. So maybe culture media is often in a gel-like consistency or liquid-like consistency or in semi-solid consistency. Mainly it contains nutrients which is used to grow the bacteria and other microorganisms through plating. So it is a source of nutrients to support the growth of all microorganisms in vitro. So under lab conditions we are able to grow the microorganisms like bacteria, and it helps in the growth and counting of microbial cells and selection of microorganisms and the survival of microorganisms up to the growth phases also need to calculate it by using this culture media. Yes. Coming to the common ingredients, so there are several substances which is involved in the ingredients of culture media. First one is peptone. So peptone, it is the main source of carbon and nitrogen. So without carbon and nitrogen, microorganism can't able to grow. So it is a main source of culture media. And coming to the beef extract. So beef extract, it is a source of all amino acids, vitamins and minerals required for the growth of microorganism. And yeast extract, it is a source of vitamin, carbon and nitrogen. And distilled water is mainly essential for the bacterial growth, and sodium chloride is for maintaining the isotonic environment. So isotonic environment is nothing but where the fluid in and out will be in balanced condition. This is termed as isotonic, and sodium chloride is necessary for maintaining the isotonic environment for the microorganism. And agar is the main solidifying agent in case of solid agar or semi-solid agar. We want to add agar in respective percentage like 0.5 percentage, 1.1 percentage of agar. Based on the percentage, the agar solidifying agent will differ from one media to another. Yes, coming to the preparation of culture media. So first one, you want to weigh the ingredients powder as on the weighing machine. So how much amount of peptone, how much amount of beef extract, yeast extract, NaOCl. So you must weigh the ingredients powder and dissolve that ingredients powder in distilled water in a conical flask. And after adding distilled water, you want to adjust the pH. So if required, you want to add the agar. So then after preparation is over, it should be sterilized in autoclave for 121 degrees Celsius for 15 minutes at 15 lbs, and place the flask in the laminar airflow, and that laminar airflow should be sterilized with 70% alcohol to avoid the contamination and to maintain in sterile condition. And the media is further cooled partially, and it is poured into a sterile petri plate for the solidification. So after 15 to 20 minutes, the agar in the media in the petri plate, it gets solidified so that it is easy for us to streak and identify the microorganism. And through several techniques like spread plate technique or streak sample technique, we can use the spreader or inoculation loop for spreading and streaking. And the petri plate should be sealed with parafilm, and it should be labeled like what is the date and what are the purpose and which microorganism is involved for the growth. Like that, several of the details were labeled in them. And this should be incubated inverted position. So while plating is here, we want to invert it up and down for 24 hours at 37 degrees Celsius and observe any colony formation on the next day. So for bacteria, 24 hours incubation is absolutely necessary, and it's sufficient for the growth of bacteria about 24 hours. So after parafilm pack and label, we want to invert the petri plate into the incubator, and several colonies were observed in that plate on the next day.